Okay, welcome everybody. And this is chapter 8 on gene expression and control. And what we're going to address today is how it is that the information molecule is used to produce the tools of life when and where the cell needs it. Okay, remember that the tools of life are proteins, and the information for the tools of life are in information blocks in DNA called genes. Okay, so we're going to talk about what a gene is and how it's used. And then we're going to talk about transcription and translation, where they happen and what they're for. And we'll talk about the genetic code. And then we're going to try to do some examples of transcription and translation. We'll talk about mutations, why gene expression is necessary, and talk about a little bit about genetic engineering. So the ability to make proteins is vital for the survival of an organism, right? Remember, we've talked about the fact that since proteins are the tools of life, then if you can't make protein, you can't survive, okay? Some examples, right? Seeds of the castor oil plant contain a protein called ricin, which is a deadly poison that inactivates ribosomes that assemble proteins. It's been used by terrorists as a bioweapon, okay? Other ribosome inhibiting proteins are more deadly, such as abrin from Jaquarty beans and the Shiga toxin from Shigella bacteria. Why are these tools out there? They're used as defenses by these organisms against attack, okay? But they can be engineered to obviously other purposes, right? You can see some examples of each of these here, right? On top there is the castor oil plant, the source of the ribosome busting ricin, and below is the umbrella modified to fire a tiny pellet of ricin into the victim, right? This was used to assassinate Georgy Markov in the streets of London in '78, right? So pretty nasty stuff, right? All right. What is a gene? A gene is a packet of information for a particular tool of life, all right? The job of DNA is to contain and maintain the information needed to build the tools of life, the proteins. But the other job of DNA is to know when and where that information needs to be used, okay? And that's gene regulation. Transcription is the process of enzymes using the gene's DNA sequence as a template to assemble a strand of messenger RNA. Translation is where we use that messenger RNA and build a sequence of amino acids, a protein from that, all right? The result is the tool that the cell needs in order to function, all right? Now, important point here. Remember that we talked earlier about the fact that there's two basic types of cells, right? There are prokaryotic cells and eukaryotic cells. In prokaryotic cells, transcription and translation happen simultaneously in the cytoplasm. In eukaryotic cells, transcription happens in the nucleus and translation happens in the cytoplasm. So it's a very important distinction to make, okay? Let's take a look at DNA and RNA and revisit some of these things. And let's make sure that we got the sound the way we want it. So hold on.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these three bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of two polynucleotide chains. The two DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The two polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
So what we see with DNA, right, is a double-stranded molecule that has complementarity. All right. We talked in the previous chapter about the fact that everywhere on a given strand that there's an A, on the opposite strand there's a T, and everywhere on that same given strand there's a G, on the opposite strand is a C. And when DNA is reproduced, the double helix unwinds and the information in the old strand is used to make the new strand based on those base pairing rules. And the result at the end of the process is two double-stranded DNA molecules with the same information in them, unless there's been a mutation. And a mutation can have one of three outcomes. It can be good or bad or neutral, right? A good mutation means that the organism is more likely to live long enough to breed. A bad mutation means the organism is less likely to live long enough to breed. And a neutral mutation means that there's no real effect, okay? RNA is used to bring the information from the DNA to the ribosomes, all right, which use that information to build the protein in the process called translation. The type of RNA that does that is called messenger RNA, all right? Transfer RNA is used to bring amino acids to the ribosomes so that they can be incorporated into the polypeptide chain. And ribosomal RNA, which is produced in the nucleolus, is a structural component of the ribosome, all right? So 3 roles for RNA. There are actually more, all right? There are RNAs that regulate gene expression. We'll talk about those in a little bit later, all right? Gene expression itself is the use of those information blocks in the DNA to make the tools of life when and where the cell needs them, all right? It doesn't make any sense to produce a tool when you don't need it. And it doesn't make any sense to not produce a tool when you need it. So some genes code for proteins that are in use pretty much all the time, and they're called constitutive. And then some genes code for proteins that are needed in specific situations, all right? And those have patterns of expression that can be specific to a particular tissue or a particular set of conditions. And those conditions usually act on a part of the gene known as the promoter, which is the gatekeeper to the rest of the information in the gene, okay? So gene expression is simply the process by which the information in a gene becomes converted to an RNA or a protein product, okay? Let's take a look at transcription, which is taking DNA to RNA.
Transcription is the process in which DNA is used as a template to produce a type of RNA called messenger RNA or mRNA. If you imagine our genome, that is, all the genes in our DNA, as a book of recipes in which each recipe is a gene that carries out the instructions on how to synthesize proteins, then transcription copies one of those recipes from the cookbook onto a Post-it note. The Post-it note with the copied recipe is the messenger RNA. Recall that DNA and RNA have a similar way of encoding information using a sequence of 4 different nucleotide bases. For DNA, those bases are A, C, G, and T. RNA has the same A, C, and G, but it contains the base U instead of T. DNA and mRNA are complementary to each other. This means that the nucleotide bases C and G always pair together, and A always pairs with T in DNA and U in RNA. Messenger RNA carries the same information as DNA, but transcribed in the code sequence of RNA. The key enzyme that carries out transcription by reading the DNA template and building a complementary mRNA is called RNA polymerase. The name is easy to remember because you know that the enzyme synthesizes RNA and that a polymer is a long chain of subunits, in this case, a long chain of nucleotides. Now let's focus on the 3 stages of transcription: initiation, elongation, and termination. Initiation begins when proteins called transcription factors bind to the promoter, a special sequence of nucleotides on the DNA strand that indicates the starting point for RNA synthesis. Next, RNA polymerase binds to the promoter, and a complex of enzymes, including helicase, unwinds the double helix and stabilizes the individual strands of DNA. Elongation proceeds as RNA polymerase attaches one nucleotide at a time to build an mRNA transcript complementary to the DNA template strand. RNA polymerase can elongate the growing RNA strand at speeds faster than 60 nucleotides per second. Elongation is terminated when RNA polymerase encounters a specific terminator sequence on DNA that signals the end of the gene. At this point, enzymes catalyze the release of the mRNA transcript, and the completed mRNA transcript detaches from the RNA polymerase.
What happens next, all right, is that message is going to be processed. Sequences that don't code for amino acids are going to be removed in a process called splicing, all right? There's going to be a poly A tail put onto the 3' end of the message by an enzyme that's able to do that without using a primer or a complementary strand, all right? And then there's going to be a 5' chemical group called methylguanosine put on the beginning of the messenger RNA. Now, why does that happen? That happens in eukaryotes so that the ribosomes that are going to use it for translation know where to start and where to stop, okay? And it also determines, in part, how long the message is going to last out there in the cytoplasm, okay? Transcription is similar to DNA replication. The DNA strand is the template for RNA production, and the base pairing rules apply, except for the fact that there's uracil instead of thymine in RNA. RNA polymerase adds the nucleotides to the end of the growing strand, and we generate the RNA molecule. So you can see here what the RNA molecule looks like. And typical RNA molecule, which is single-stranded up top. DNA molecule is double-stranded, okay? Both DNA and RNA are polymers of nucleotides. One base differs between DNA and RNA. Uracil occurs only in RNA, and thymine in DNA. The sugar in an RNA nucleotide is called ribose, and the sugar in DNA is deoxyribose. Ribose has an additional oxygen on it, where deoxyribose doesn't have that, right? And that's one of the things that actually makes RNA less stable and less long-lived than deoxyribose, okay? And the reason for that is that a chemical reaction can occur where RNA can actually break itself down in water under specific circumstances, and that chemical reaction doesn't occur with DNA because it doesn't have that extra oxygen, okay? In transcription, RNA polymerase binds to a region of the gene called the promoter, which acts like a gatekeeper to the rest of the information in the gene. The polymerase moves along the DNA and unwinds the DNA so it can read the base sequence. RNA polymerase incorporates the RNA nucleotides in the order determined by the sequence of the gene on the DNA strand. The new messenger RNA is a copy of the gene from which it was transcribed. So if we look here, right, you can see that you've got two strands in the DNA, the coding and the non-coding strand, all right? They're complementary, all right? This is just a diagram. Remember that this is going to be double helical in the actual molecule, right? And then during transcription, all right, the non-coding DNA strand is a template for the RNA synthesis, okay? And we make a copy of that. And then that new RNA, which is complementary to the non-coding strand, is going to code for the amino acids that are going to be incorporated into that particular protein, okay? Now, there are modifications that take place in eukaryotes before it can be used in translation. And we talked about some of these already. The introns, which are the pieces of information in the gene that do not code for proteins, are removed in a process called splicing, and that happens in the nucleus. And then the exons are stitched together, all right? And then a poly A tail is added to the 3' end, and a 5' methylguanosine cap is added to the 5' end. Now, this is only in eukaryotes, okay? Prokaryotes are a bit different, right? So you can see here the process of transcription, right? Zooming in. Remember that what we call the nascent RNA, the freshly made RNA, doesn't have any of those modifications yet and can't be used for translation, right? It hasn't been properly prepped. And so those things in the nucleus that we talked about, splicing, poly A tail, capping, have to happen before we're really good to go and can leave the nucleus through the nuclear pore, all right? And then a ribosome can latch onto it and start reading it, okay? The information in messenger RNA consists of 3 nucleotides that form words spelled with the bases A, C, G, and U. 64 codons constitute the genetic code. Most codons specify amino acids, but there are a few of them that are called stop codons, which are an indication to the ribosome to quit translating and to disengage from the message and then release the protein, okay? The sequence of 3 nucleotides in a base triplet determines which amino acid the codon specifies, and the order of codons in the messenger RNA determines the order of amino acids in the polypeptide that are going to be translated from it, okay? 20 amino acids are encoded by 64 codons in the genetic code. Some amino acids are specified by more than one codon. Other codons signal the beginning and the end of a protein coding sequence. Most organisms use the same code. So let's look at transcription to translation here.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of the messenger RNA, called the sigma factor, participates in recognition of the promoter region. Soon after transcription is initiated, the sigma factor dissociates from the RNA polymerase. RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches a specific sequence of nucleotides on the DNA, called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA.
Translation is the process by which the information contained in a molecule of messenger RNA is used to synthesize a protein. The process is called translation because ribosomes read along a messenger RNA molecule, translating the genetic code into the amino acid sequence of the protein. The genetic code contains 61 sense codons, codons that code for an amino acid. However, these 61 sense codons only code for 20 amino acids. Therefore, most amino acids are coded for by several codons. In other words, in the language of the genetic code, there are 2 words for phenylalanine, 6 for leucine, 3 for isoleucine, and so on. This overlap of meaning is called degeneracy. The degenerate nature of the genetic code means that mutations in DNA do not necessarily result in mutations in the resulting protein. The first stage of translation is called initiation. During initiation, the 2 ribosomal subunits, a tRNA with an anticodon complementary to the start codon, and several protein factors assemble on the mRNA at the start codon. This results in an initiation complex ready for translation. During elongation, the tRNA molecule with an anticodon corresponding to the next codon enters the A site. The ribosome catalyzes the transfer of the amino acid or polypeptide on the tRNA in the P site to the amino acid on the tRNA in the A site. After the formation of this peptide bond, the ribosome moves along the mRNA to the next codon and repeats the process. When the ribosome encounters a stop codon, a protein called a release factor binds in the A site, halting elongation. This process is called termination. The ribosome separates into its 2 subunits, and the mRNA and newly synthesized polypeptide are released. The ribosomes can then be recycled to continue the process.
Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. The initiating transfer RNA carrying its amino acid binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of 1 codon, and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site, and the ribosome shifts down by a distance of 1 codon. This is followed by the 2 amino acids on the transfer RNA in the P site being transferred to the 3rd amino acid. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.
And so that's your overview. Okay. Transcription in the nucleus in eukaryotes, translation in the cytoplasm or on the rough endoplasmic reticulum in eukaryotes. And then once the protein is manufactured, it ends up being loaded from the rough ER into the Golgi, which then adds chemical groups to it. The Golgi then ships those proteins out to their final destination in secretory vesicles, and it starts to do its job. Okay. So that's the flow of information in a eukaryotic cell. Okay. So when we're talking about transcription to translation, okay, we're talking about an exact copy of what's in the DNA, except that we're using uracil as one of the letters in the alphabet rather than thymine. All right. So it's an exact copy. So what we refer to as the information that builds the protein is called the reading frame. Okay. And so let's write that down. Okay. Reading frame. So reading frame. Okay. And to get to the reading frame, right, we have our new, or we call that nascent RNA. Okay, which is going to contain combinations of exons and introns, right? We'll just make our gene a very simple one here. Okay. The exons can be of various sizes. The introns can be of various sizes, okay? So that's our nascent RNA, okay? There's usually what we call a 5' untranslated region and a 3' untranslated region. Okay. So this is again all in the nucleus. Okay. The next thing that happens is that the exon, we'll just number the exons. 1, 2, 3, and 4. Okay. The exons are spliced together. Okay. So they get knitted together. Right. 1, 2, 3, and 4. Right. With again a 3' and a 5' UTR. Right. And then we have capping, and polyadenylation. PolyA tail. Okay. And then where do we end up? We end up with something that looks like this. We'll put the cap here like that. And we'll do 1, 2, 3, 4. And then another untranslated. And then we've got the polyA tail coming off the end. Okay. This is the 5' cap. There's the 3' tail. Okay. And then these little boxes that are stitched together, they contain the reading frame. Okay. The reading frame is the sequence of codons used to build the protein. Okay. And that's why you have to stitch together all of these exons, right? That's an exon. That's an exon. That's an exon. And that's an exon, right? You have to stitch all of those together to make the reading frame, right? Then you have to modify it. Then what happens? Well, we leave the nucleus through pore. Okay. And then in the cytoplasm, or rough ER, ribosome binds. Five prime end. Okay. And then we have translation. Okay. Right there. 3 types of RNA are involved in translation. The messenger RNA has the information, right? The ribosomal RNA is a structural component of the ribosome, and the transfer RNA delivers the amino acids to the ribosome. Ribosomes have a large and a small subunit, okay? Right? There's the large subunit, there's the small subunit. And the message would go between them, okay? That would be the messenger RNA. Okay. So like a kind of like a hot dog bun and a hot dog. Okay. Transfer RNA has an area called the anticodon, right? And the message is going to be complementary to the anticodon. So the anticodon for this one, for instance, would be AUG, right? This would be the messenger RNA, right? And the amino acid would be up here, okay? So amino acid attached. Okay. So this is how we know as a ribosome which transfer RNA to grab, right? Because what will happen is that the ribosome checks for goodness of fit before it moves to the next codon. If the fit's not good, then it's the wrong transfer RNA, right? Translation occurs in the cytoplasm of all cells, okay? Messenger RNA is transcribed in the nucleus in eukaryotes. Okay. In the cytoplasm, the small ribosomal subunit binds to messenger RNA. Initiator transfer RNA uses its anticodon to pair with the first messenger RNA codon. The large ribosomal subunit then slaps onto that assembly, and then the ribosome slides down the message one codon at a time, incorporating the appropriate transfer RNA with its amino acid attached into the growing protein. And this ends when we get to a stop codon, right? So you can see here how we set this up, right? We've got the production again of the message. See how it's leaving through the nuclear pore, right? Okay. Transported to the cytoplasm. And over here, what do we have? We have the cap would be up here, 5' cap. Okay. And the end of the message, the poly-A tail will be over here. Okay. Okay. So 5' to 3'. All right. The transfer RNAs carry their appropriate amino acids. And as we walk from codon to codon down the reading frame, we incorporate that into the protein gets assembled. Okay. You can see here, right? Translation begins when the ribosomal subunits and the transfer RNA converge onto the start codon of a messenger RNA, which carries a methionine. All right, that is the signal to begin. Okay, and the methionine codon that does that is always contained within a context of sequence that tells the ribosome to freeze there and stop and begin translation. Okay, there might be other codons along the way from the cap to the beginning of the reading frame, but they're not part of the reading frame really, okay, because they don't have the proper set of nucleotides around them to tell the ribosome to stop. Okay, in eukaryotes, that area to stop is called the Kozak sequence, Kozak. Now, this is not in your book, all right, but it just serves as a braking mechanism for the ribosome to stop where it's supposed to. Okay, so it doesn't start translating in the wrong spot, right? And then we start the process of translation, right? The methionine gets incorporated, then we have the next amino acid that's coded for get incorporated, then they get hooked together by enzyme activity that's part of the ribosome, and then we slide over another set of nucleotides and we eject the transfer RNA that no longer has its amino acid attached to it, right? That's in the E site, the eject site, right? And then we move on and on and on and on until we get where? Until we get to the stop codon, right? And at the stop codon, we don't code for a transfer RNA that contains an amino acid. We instead encode for a transfer RNA that contains a water molecule. And that is the indication to quit, okay? So we release the protein, okay? We release the protein, the two subunits disassemble, right? And then if we want, we can start all over again, all right? Now, how long does the message last? How long do we do this? Okay. And one of the main factors is until the poly-A tail is gone. Okay, that's one of the factors. All right. Once the poly-A tail is gone, the message gets broken down, and then we're kind of done with this. Okay. So you can see here again, how we go, right? Around and around and around. Okay. Mutations are permanent changes in the nucleotide sequence of DNA, which can change a gene product. Okay. It can be a good or a bad or a neutral change. And here we're referring to reproductive success, right? A mutation that changes a gene's product can have a harmful effect. One example, mutations that affect proteins in hemoglobin can reduce the blood's ability to carry oxygen. And an example of that is sickle cell anemia. Okay. Types of mutations, not in terms of, the result of the mutation, but what sort of change takes place in the actual message. Can be a base pair change, okay? Can be an insertion, can be a deletion, all right? Mutations that shift the reading frame of the messenger RNA can result from the deletion of one or more base pairs, the insertion of one or more base pairs, okay? And the example can be beta thalassemia, if it happens, for instance, in a hemoglobin gene, all right? What causes mutations? Well, viruses, transposons, radiation, and mutagens, chemicals. And then finally, regular mistakes in DNA replication and transcription. Because, nothing's perfect, right? Example, right? Hemoglobin, which is the oxygen binding protein in red blood cells, which is a molecule that carries oxygen inside red blood cells around the body, is made up of 2 alpha and 2 beta globins. Each globin has a pocket that holds a heme, which holds an oxygen, to an iron that's in the middle of the heme, okay? And if you have a particular type of mutation that changes a glutamate to a valine, all right, this would be the normal beta chain of hemoglobin. And this is the sickle beta chain of hemoglobin. This is a mutant. Okay. Then what happens is under low oxygen concentration, that particular type of beta chain, the mutant, will polymerize, and that will cause red blood cells that carry enough of it to take on a crescent shape or sickle shape. And the result then is it can't carry oxygen as effectively. Okay. So there you go. Right. So that was a single substitution. Right. An insertion will add a nucleotide into the middle of a reading frame, and that can result in a shift for the rest of the message. The resulting protein may be too short and not assemble correctly. An example here is in beta thalassemia, right? We got an insertion in there, and the result is that we prematurely stop building the beta chain, and the result then is that we have defective hemoglobin, right? So a deletion or an insertion, right? These are 2 different mutants, okay? That's beta thalassemia. Okay, anytime you see emia, that means in blood. Okay. All the cells in your body carry the same DNA, except for lymphocytes. And gametes. Okay. A typical cell in your body uses only about 10% of its genes. Differentiation is the process by which the cells become specialized and occurs as different cell lineages begin to express different subsets of their genes. A transcription factor is simply a protein that says when and where a particular gene is going to be used. All right. So here we kind of have to go back in time a little bit. Okay. So let's do a whiteboard here. Let's do a new share. Let's do a whiteboard. Okay. And let's do a little sketching here for just a second. Okay. So when we build a new human being, right, we have an egg. There's a nucleus, and we have a sperm cell, right? Right. The egg is haploid, and the sperm cell is haploid. Okay. And they come together in a process called fertilization. And you make a zygote, okay, which is diploid. Okay. It's a zygote. Right? Haploid is designated as little n. Diploid is designated as 2n. Okay? All right. The next thing that happens is cleavage. Okay? One cell becomes two cells become four become eight becomes 16, and so on. And eventually we get to this thing called a morula. Okay? This is all happening in the fallopian tube of the female. Okay? The morula works its way down the fallopian tube and eventually becomes something called a blastocyst. Okay? This will be the inner cell mass. That's going to become U eventually. And out here is what's called the trophoblast. Nuclei in there. Okay. Blastocyst. Okay. And that right there is the inner cell mass. Right? And that is going to drift into the uterus and implant. Okay. And then that will develop into a gastrula. Okay. And the gastrula is going to look sort of like this. Okay. The gastrula is going to have an embryonic disc in the middle. That's going to be you. On top is going to be a sac called the amnion. Below it is the yolk sac. Okay. And around the whole thing is a membrane called the chorion. Okay. And that's the embryonic disc. Okay. And then if we take that embryonic disc and we take a cross section through it and look at it, all right, at some point, there's going to be the development of 3 layers of cells. Okay. And there's a little structure here called notochord, right? So on the top, let's make a different color to the top. There you go. Let's go with, I don't know, yellow, kind of tough to see. Purple, right? Up top is going to be ectoderm. Okay. And underneath is going to be endoderm. You got it in orange. Okay. And in the middle is going to be mesoderm. Do that in red. Okay. All right. What's the point, right? Well, mesoderm and ectoderm and endoderm become different things, right? Endoderm becomes the inner lining of GI, respiratory, urinary, and reproductive. Sorry, it's tough to read. Okay. Ectoderm becomes the epithelia of your skin. And mucous membranes, as well as your nervous system. Okay. And mesoderm becomes everything else. Okay. How does that happen? Well, there's 3 processes. Okay. Well, 4. Okay. One is mitosis. Another is growth. Another is morphogenetic movement. And then finally, we have differentiation. Okay. Now, this is a process, overall process is called development. Okay. How do these processes happen? All right. Well, mitosis is cell division, and that's fairly easy to understand, right? Growth is an increase in size. That's easy to understand. Morphogenetic movement is the movement of one population of cells from one location to another, all right? And that follows chemical cues, right? So you follow a chemical trail and you literally walk from one location to another location using ATP for energy, okay? What is differentiation? Well, that's where we get into gene expression, okay? So let's clear all of this. Well, here, clear all the right. There we go. That's what we want. Okay. All right. So with differentiation, okay, a cell starts out as a totipotent stem cell. Which means could become anything. Okay. Then it becomes a pluripotent stem cell. Can become several things. And then finally, it becomes a differentiated cell. Does one thing. Now, the genetic material at every step, right, is the same. There's been no changes to the genetic material in this process. So why the different designations here? And the answer is because of the way that the DNA gets used, okay? At each step along this pathway, different genes are rendered inaccessible to transcription, okay? So you lose access to some genes, okay? And that's through chemical modification, all right? We add methyl groups to them, for instance, and then you can't use them to make messenger RNA from anymore, all right? And then as you go through time and space a little further from a totipotent stem cell, in the process is differentiation, sorry, and then this should be 1, 2, 3. All right, 1, 2, and 3. Sorry about that. 1, 2, 3. Okay. So you lose access to some genes. All right, when you go from a totipotent to a pluripotent stem cell. Then you lose access to more genes. Okay. By chemically modifying them. And then finally, you're the differentiated cell and you only use genes you need. Okay. So because of this, development only goes in one direction. It goes from general to specific. Okay. Because of the way the information gets modified. So what are the mechanics of that? You encounter a chemical signal in the environment that binds to a receptor on the or in the cell, and that leads to modification of certain genes so that you can't use them anymore. Okay, and probably modification of certain genes so you can use them more. All right. So the result is the information's all still there, but what you can read from is different. Okay. And if you can only use certain subsets of genes, then that's going to direct what you become. Okay. So as an example, you start out as a totipotent stem cell, you can read all your DNA. As a pluripotent stem cell, maybe you end up now with possible fate of becoming, say, muscle or bone or connective tissue. Okay. And then finally, as time goes by, you can only read, say, from the muscle cell handbook of DNA in your nucleus. And that's it. That's what you're going to be. Okay. The other information's there, but the other information can't be accessed. Okay. You can't go back in time and be something else. All right. So that's what drives it. Okay. That's why when you begin development, when the sperm meets the egg at fertilization, you have to have the right number of genes to make the right number of proteins in the right place at the right time in order to build a new organism correctly. Okay. So that's why if you have improper chromosome number, for instance, or you have chromosomal deletions or or insertions or whatever, your information's messed up early on, you're going to have big problems that may not lead to the proper completion of the process. Okay, so that's kind of the overview of it. Now, your book probably doesn't get that deep into this, but I wanted to give you kind of an overview of why this matters and how it happens. Okay, so let's go back to where we were. Share that. All right. So transcription factors are proteins that influence the transcription by binding to DNA. Okay. A master gene is a gene that encodes a product that affects the expression of many other genes. Okay. And controls many tasks, such as the production of, say, an eye or a particular bone or some other structure. And a homeotic gene is a type of master gene that controls the formation of specific body parts during development. Okay, so the way that they're envisioning this, okay, is that you have at the top of this a master gene that talks to downstream genes that talk to specific genes okay, that lead to a particular structure being built. Okay, so master gene encodes a protein or proteins. Okay, they affect the use of these downstream genes because they act as transcription factors. Those proteins that they encode control specific genes, and then that leads to the production of a particular structure. So if we change here, we're going to lead to changes here and here as well, and finally here. Okay. Researchers are studying the function of homeotic genes by changing their expression, introducing mutations, or deleting it entirely. We can take DNA and do that, right? We can take DNA from a source. We can, through the use of various tools, remove pieces of the information, add other pieces of information, and then put that altered information back into the organism. There's ways to do that, and then see what happens. Okay. Examples include things like Antennapedia, Tin Man, and Groucho. Those are just nicknames for these homeotic genes that they've worked on. A lot of homeotic genes can be interchangeable within a species. Okay. Example, PAC6 in humans is the eyeless gene in flies, right? And what you're seeing here, right, is a normal fruit fly on the left with round eyes. A fly with a mutation in its PAC6 gene, its eyeless gene, develops without any eyes. Okay, so obviously that gene is way, way high up on the cascade of events that leads to the production of an eye. Okay, so very important that you understand that there's a sequence of events that leads to control, right? So there's a first gene, then there's a second set of genes and a third set of genes, and so on and so forth. Okay, and depending on where we are in the organism, all right, that first gene may be different, all right, in those regions, and the second genes may be different, and so on. Okay. In humans, many other animals, the PAC6 gene can affect eye formation. In panel B, eyes form wherever the eyeless gene is expressed in the fly embryo, here on the head, and also on the wing of this fly. The PAC6 gene in humans and other animals is so similar to eyeless that it triggers the eye development in fruit flies. So they're using the human gene in the fly here to cause that to happen. A normal human eye has a colored iris surrounding the pupil, Mutations in the PAX6 gene in humans cause the eyes to develop without an iris, a condition known as aniridia. Okay. Now, you might say, well, who did that? No, that was a naturally occurring mutation that they were able to find in people with that condition. And then they knew that that was in the PAX6 gene. And so they studied the effect in the model organism, the fly, and they figured out that that's what was going on. Okay. No one's experimenting on human genetic engineering for the purposes of basic research, at least not here in the US. Okay. In mammals, males have only one X chromosome and females have two, but one is usually condensed in what's called a bar body and not expressed. According to the theory of dosage compensation, X chromosome inactivation equalizes the expression of X chromosome genes between the sexes, such that males always use their X in every cell in their body, and the Y, of course, only has the trait of being male. And in females, you use one or other X. Which one you use is what's called stochastic or random. Now, why would this be the case? Again, we go back to this idea of gene dosage, right? You have to have the right amount of information to make the right amount of proteins, to make the right amount of tools of life for the organism to function normally, right? And if we had two X's firing at the same time, we would have a gene overdose, and that would probably alter the function of the cell, okay? So all females, all diploid females are what are called mosaics, meaning that they are using one or other X chromosome as a method of expressing those particular genes, okay? Whereas guys only use the one, okay? The Y chromosome is different, right? The Y chromosome has one trait on it, all right? The trait of being male, all right? X chromosome, about 2,000 genes that we know so far. The Y chromosome, about a little under 500, okay? It has the master gene for sex determination, triggering testes formation. Testosterone made by the testes controls the formation of secondary sex characteristics, okay? Stuff other than the gonads, the ovaries and the testes. The absence of the SRY gene in females triggers the development of ovaries and female characteristics, okay? So the only trait on the Y chromosome is the trait of being male. How many genes play into that trait? Oh, more than one, obviously, okay? The human X chromosome, though, has genes that play into not only gender, but other body plan information. And we know that because there are sex-linked diseases, mutations on the X chromosome, that affect body systems other than the reproductive system, okay? Such as color blindness and Duchenne muscular dystrophy and hemophilia, all right, just to name a few. Lactose intolerance is an example of an inherited disorder, all right? Most species lose the ability to break down lactose, which is a disaccharide in milk, after a certain age. In humans, this usually occurs around age 5. The lactase gene is no longer utilized. Once lactase production slows down, lactose passes undigested through the small intestine in these individuals. Bacteria in the large intestine begin to break it down, producing gas and pain and distension, and that's called lactose intolerance. About 1/3 of human adults have a mutation that allow them to digest milk throughout life, okay? Transcription is affected by chromosome structure. We talked about this earlier when we were talking about development. Modifications that can change gene expression include adding a methyl group to a histone protein or methylating the DNA directly, and that can produce something called heterochromatin. Let's write that down. And what is that? That's untranscribable DNA. Okay. Once the nucleotide gets methylated, it stays methylated in the cell's descendants, and that affects its use in everything that derives from that modification. So all cells that are descendant of those methylated genes are going to have those genes methylated too. Okay. And this leads to a discussion of epigenetics. All right. Methylation of the chromosomes is normally reset in the first cell of a new individual. All parental methyl groups are not removed, so some methylations can be passed to offspring. Boys are affected by the lifestyle of individuals in the father's line, girls by individuals in the mother's line. Okay. Heritable changes in gene expression that are not due to changes in underlying DNA sequence are what are called epigenetic. Okay. Epi meaning above, genetic. Okay. So this is sort of like a second genetic code. Okay. The methylation pattern can determine which genes are used and which aren't. All right. That expresses that has an effect on the expression of those genes, and that results in a change in the way the organism develops. Okay. And so this is one of the reasons why we believe now that the mother has more effect on gene expression than the father does, because the mother has the methylase enzymes that act on the DNA that comes from both the mother and the father. All right. And so very often her epigenetics can be more powerful than the father's epigenetics. So mom has more of a say in how you turn out than dad does. Okay. And that's just because she's giving you all her cytoplasm and of course, all of her mitochondria. You get all of your mitochondria from your mother. Okay. And as a result, that can have an effect on certain subsets of genes. All right. Okay. Why is it advantageous for the genetic code to be redundant? Okay. Does ricin impact transcription or translation? How does it work? And how are household cleaners and grocery foods related to mutations in our DNA? You might want to, Google some of those things and think about the questions that they raise. And if you have questions about that, we can talk about them in class. Okay. All right. So that brings us to the end of this discussion, and I will see everybody in class next week.